Whether you're Martian or human, it's not until you step inside a building that you're likely to understand what its function is, what goes on there, and how it's done. What would a Martian make of this business? Here are machines transforming this into this. But what are these machines actually doing? Well, to us humans in the know, it's simply a newspaper print works. But more significantly, each machine is doing a specific job as part of the overall process. And this is worth analysing more carefully, as it has much in common with cellular processes. If you're going to produce millions of newspapers a day, every day of the year, you need an efficient system. The key is a production line, a stepwise process with the products of one step being passed on to the next in the sequence as quickly and smoothly as possible. And each machine in the production line acts very much like a catalyst. Each does a specific job very quickly, much more quickly than we could do it unaided. The line doesn't even have to be stopped to load a new roll of paper. At full lick, this line can turn out 60,000 papers each hour. Even the quality control checks are aided by remote control. The speed of the printing press and the density of ink in each column is checked and recorded on computer, and automatic adjustment is made. Similarly, with alignment of the print, everything is registered and controlled by a computer. Compared to our own abilities, the power, speed, and accuracy of the process are awesome. How many ton rolls of paper could you lift? How quickly could you count and bundle 125 copies? How many monks with quill pens would it take to match this monster? So what makes the whole process possible is a series of specific machines, each speeding up just one particular step, and each machine connected to the next to form a production line, a defined pathway. So how do these features apply to the cell? A living cell is also like a factory, or more like a group of factories, the cellular compartments or organelles. And if we could step inside one of these, we should find a number of cellular production lines, the metabolic pathways. Each step need not be physically linked to the next, but substances are broken down or synthesized in stepwise sequence in these pathways. And the cellular machines that speed up, catalyze each step are very specific in their activity. They're the enzymes. Enzymes outperform by far the catalysts prepared by chemists, and as enzymologist originally trained as a chemist Alan Furst readily admits,
enzymes have long been admired by chemists for the power of their catalysis and their specificity. Enzymes can choose one molecule out of a whole soup and act just only on that. They can speed up a chemical reaction by a factor of perhaps 10 to the 12th. An enzyme can just use one part of a molecule to react upon and leave the rest of it aside. A chemist wishes he would be able to do this himself.
So what endows enzymes with these remarkable properties of specificity and catalytic power? We'll focus on just one enzyme to explain what has been discovered about these mechanisms to date. Our enzyme is just one in the pathway that leads from glucose to pyruvate, a 10-step pathway known as glycolysis. Phosphofructokinase, or PFK for short, catalyzes the conversion of the substrate, fructose 6-phosphate, to fructose 1,6-bisphosphate. The additional phosphate is donated by a second substrate, ATP, converting it to ADP. And this is the subject for our consideration of this catalytic mechanism, an accurate computer model of PFK based on crystallographic measurements. But first, crystals of the pure enzyme must be isolated from the bacteria in which PFK occurs. The bacteria are grown in a special medium, which is kept aerated overnight by a steady shaking motion at a constant temperature of around 35 degrees centigrade. They're genetically engineered bacteria, designed to produce a high level of PFK, thus making its isolation and purification relatively easy. By the next morning, a thick suspension of bacteria has been produced. This is then centrifuged, and the pelleted bacteria are then resuspended in buffer. To obtain PFK, we've to break open the bacteria. Packing the suspension in ice prevents thermal denaturation of the cellular proteins. The bacteria are then subjected to ultrasound. This sonication disrupts the bacterial cell walls, releasing the cell's contents, including PFK, into the buffer. Further centrifugation, at higher speed, removes the cell debris, and the impure PFK is left in the supernatant. The final stage in the purification of the enzyme is to pass the supernatant through a chromatographic column that is specially designed to retain just the PFK, while letting the other proteins pass through. The stuck PFK is then removed from the column by washing through with a suitable eluent. The fractions, including those containing the PFK, are collected in tubes by means of a fraction collector, a device that counts and collects drops. Now we've got pure PFK, we're a little closer to being able to work out how the catalysis is achieved. But what sort of information do we need to obtain? We spoke to a Cambridge scientist who has worked on various aspects of PFK for 14 years, Phil Evans.
Well, to understand the way an enzyme catalyzes its reaction, we really need to know its structure. We need to know the arrangement and space of the groups on the enzyme which perform the catalysis. X-ray crystallography is currently the only way for most proteins of getting this information, although it is maybe a laborious way of doing it. An enzymologist once said that a picture of a horse doesn't tell you how fast it can run. On the other hand, if you didn't know that the horse had 4 legs, you'd find it very difficult to understand how it was doing its running.
X-ray crystallography shows us that our horse, PFK, has 4 legs, or more accurately, 4 subunits. They're identical, but they've been coloured differently for clarity. 2 shades of yellow and 2 shades of blue. There are 4 active sites where catalysis occurs. As the model rotates, we can see substrates bound in 2 of these sites. The purple shape is fructose 6-phosphate, and the orange-red shape is ATP. Just as this model of the surface has been created from X-ray crystallographic data, the detailed molecular structure also can be deduced from such data.
The structure of the protein, the structure of the enzyme, and the structure of its interactions with the ligands, what groups are involved in binding with substrates, provides the framework on which you can hang all the solution information, the kinetics, the binding information, and all the information about the chemistry of the enzyme is hung on the structural basis. Which groups on the enzyme are close to which groups on the substrate. And this can tell us which groups are likely to be catalytic and are involved in binding, and distinguish those groups which are directly involved in the catalysis or binding from those that are only indirectly involved.
But how does one actually get the data and use it to construct such models? The starting point is to take the solution of purified PFK and grow a crystal from it. But getting the crystal can be quite complicated. Here, the crystals have been grown in a vessel sealed with a fine membrane. Once they've reached a suitable size, one of them is selected for X-ray crystallographic measurement. The crystal is drawn up into a fine tube with a little liquid to keep it moist. The ends of the tube are then sealed with wax, and it's ready for the X-ray machine.
In the simplest case, where you're looking at a protein where a closely related one is known, starting with good crystals, it's possible to get a structure out in a matter of weeks. But if you have to start for a protein in solution and grow crystals, it can take anything from months to years, depending on how lucky you are. There's a good deal of luck in this business.
The crystal is mounted on the X-ray machine and bombarded with X-rays. The tube and the liquid droplets are not crystalline, and they do not show up in the X-ray diffraction pattern on the monitor. Each crystal may have 10 to the 15 molecules in it. But by rotating the stage slowly and taking diffraction patterns at literally thousands of different angles, it's possible to average the data and define the structure of a single PFK molecule. With powerful computation, this wealth of diffraction data can be displayed as electron density contours of the atoms that make up the enzyme molecule.
We've gone through periods when a lot of results were coming out very quickly, and other times where things seem to be very frustrating and nothing's worked for a long time. And I imagine it's the same in other branches of science.
Here, the electron density map of the molecule is displayed at different orientations. Chemical analysis has also revealed the primary structure, the amino acid sequence, of the protein. We can couple this knowledge to the contour map to find where each amino acid lies in the three-dimensional protein structure. For example, taking the known shape of a tyrosine side chain, we can see how it can be fitted to the contour map at this point. The flicker is due to the massive amount of data that the computer is handling. The model is rotated to get the best fit in the structure. As we rotate the map, you can see that a histidine has also been fitted into its correct location in the PFK structure. Bit by bit, we can assemble a detailed model of the complete enzyme with all the amino acids identified in place. But having deduced the molecular structure of the enzyme, we're now interested in how the active site works. First, we've got to locate the active site. We can do this by combining the enzyme with substrate or substrate analogues and subjecting the complex to X-ray analysis. This reveals where the substrate or analogue binds. That's the active site. It also allows us to construct a detailed model of how the substrates sit in the active site, a logical starting point for any explanation of specificity and catalysis.
Enzyme catalysis and enzyme specificity are intimately linked. They both ultimately depend on binding and binding energies. This theme is common throughout biology. One finds in an enzyme that there are regions which are apolar, there are regions which have positive charges and regions which have negative charges. And one finds that these match up with the substrate. So the substrate binds to a surface which is complementary in structure to itself.
This complementarity of substrates and enzyme can be seen in this view of the active site of PFK. The two substrates, fructose 6-phosphate and ATP, are shown as ball and spoke models. Red balls represent oxygen atoms, purple phosphorus, and white carbon. For the enzyme, a few amino acid side chains are shown in outline. But otherwise, only the surface atoms are shown as tiny coloured dots. Once again, red represents oxygen, the purple phosphorus, and the white carbons. Blue represents nitrogen, and green sulfur. Rocking the model brings different atoms in the substrate and the enzyme in and out of view. Now watch as we flash a green outline of the perimeter of the substrates. As it flashes, look how well the two surfaces, that of substrates and that of enzyme, fit each other. Note particularly how the positively charged nitrogens of the enzyme, small blue dots, surround the negatively charged phosphates of the substrates, the purple and red balls. Active site and substrates match as lock fits key, but a very complex lock and a perfectly fitting key. So much for specificity, of matching the machine to the job. But what of catalytic power? What sorts of factors might we expect?
An enzyme uses all the factors for catalysis that a chemist would use. A chemist would often catalyse a reaction by adding an acid or base. And one finds that there are acids or bases at the active sites of many enzymes. A chemist would often catalyse a reaction by adding a metal ion. One finds that metal ions are also present at the active sites of many enzymes. But in addition to all of these chemical groups, the enzyme has one outstanding feature, and that is it binds the substrate and it can use the binding energy for catalysis. And the real factor which distinguishes enzyme catalysis from chemical catalysis is the use of binding energy.
X-ray crystallography can give us a detailed model of which amino acid side chains are involved in binding substrates. This view shows ATP and fructose 6-phosphate in the active site of PFK, with amino acid side chains indicated. There is also a magnesium ion, the Mg shown in blue, a coenzyme of PFK. From other crystals of PFK, we can construct a model of the products of the reaction, ADP and fructose 1,6-bisphosphate in the active site. With the two models, before reaction and after, and knowledge of the chemistry of the reaction, we can visualize the reaction itself and get an idea of the possible roles of associated groups in the enzyme. Here we see the reaction, the transfer of phosphate from ATP to fructose 6-phosphate, played again and again. Looking at the starting point, the substrates in the active site, note how the ATP is held by the amino acid side chains, so that prior to the reaction, the phosphate is orientated towards the hydroxyl group of the fructose 6-phosphate, to which it is then transferred. This precise orientation of substrate groups is an important feature of enzyme catalysis. But we've only been able to deduce what happens during the reaction because it's much too fast for traditional X-ray diffraction techniques, so we cannot pick out any short-lived intermediates that may exist.
Recent advances in use of Lauer crystallography with synchrotron radiation sources allows the collection of diffraction data in very short times, the order of less than seconds, maybe tens of milliseconds. This means that if you can build up a high concentration of a short-lived intermediate in the crystal, then the structure of that intermediate can be determined. And this provides possibilities of dissecting reaction mechanisms where there are short-lived intermediates in much more structural detail. The problem is that you need to build up a high concentration of the intermediate, and maybe the best technique for this in systems where it's applicable is use of flash photolysis, where a high-intensity laser flash can create a large concentration of a reactive intermediate in a very short time within the crystal. And then the rapid crystallography can be used to follow what happens to that.
For PFK and other enzymes, such techniques lie largely in the future. What they will add to our knowledge of enzymes is important, as enzymes themselves are so vital to how living cells work.
Enzymes are the workhorses of the cell. They catalyse every chemical reaction that takes place in biology, from things as simple as the dissolving of carbon dioxide to reactions as complicated as the replication of the genetic material DNA. Everything you can think of is helped by an enzyme. And enzymes even form structures. Muscle, for example, has enzyme activities. One of the most exciting challenges, I think, in science today is, first of all, to understand how enzymes work, and then to harness the power of enzymes. For example, think of the chemist. The chemist wants to make new drugs. He has various problems to do. He has to sort out which of a pair of optically active forms he wants to react. But an enzyme can do this with ease. One can actually change enzymes using protein engineering and make new enzymes to catalyse new reactions. So it's a question of not just understanding for the sake of it, it's a question that we can understand enzymes and make new enzymes. As far as I'm concerned, protein engineering is the most exciting development that has ever hit enzymology, because it enables one for the first time to change the structures of existing enzymes. And this enables us to do two things. First of all, it enables us to study enzymes by the same methods that chemists have studied chemicals traditionally for many years. And from these experiments, we learn about the properties of enzymes and how to redesign them. And secondly, we then use the knowledge for redesigning enzymes. We can do things which are useful as well as being of academic interest. For example, already I know that an enzyme that goes into soap powder has been modified to make it more efficient. One can see these things being extended almost indefinitely. I think in 15 years' time, chemists will make enzymes to order to catalyse reactions.
As we study enzymes, we appreciate more and more how elegant they are, compared to even the most ingenious machines of human invention. Enzymes have massive catalytic power and precise specificity, the results of many millions of years of evolution. If enzymes are indeed the workhorses of the cell, then they are most certainly thoroughbred ones.